Hello, everyone. My name is David Bennett with ShareVault, and I want to thank you for joining us for one of our long-awaited webinars. Before we get started, first, I want to let you know that on behalf of our panelists, a link to the slide deck and webinar will be sent out via email after the webinar. Second, we'll be answering questions at the end, so be sure to put your questions into the chat window, and we'll get to those at the end of the session. Let's get started. In partnership with ShareVault and Lumanity Bioconsulting, I'm excited to introduce Jeff Bachman, PhD, and Lisa Butterfield, PhD. Jeff, Lisa, thank you both for joining us. I'm now going to pass it over to you.
Great. Thank you so much, David, Esteban, the rest of the team at ShareVault, and a special appreciation to you, Dr. Butterfield, for joining me today. Looking forward to this discussion. So again, I lead the oncology practice at Lumanity Bioconsulting, which is the biotech or early stage focus component within the overall Lumanity organization. So, Lisa.
Hi, good morning, everyone from San Francisco. I'm Lisa Butterfield. I'm an adjunct professor in microbiology and immunology at UC San Francisco, doing some part-time consulting, and started getting into cancer vaccination in about 1995. So I'm excited to work with you, Jeff, and to present some discussion over this a topic that has waxed and waned over the ensuing years. Thank you.
Great. Thank you. Look forward to this. So how this is going to work, I'm going to go through some of my slides very quickly. Don't worry about taking notes or screenshots. You'll get access to them after the webinar. And I, in fact, have a fuller slide set. If anyone wants to email me, you can get those. And you might be wondering why I'm doing that and not Lisa, because I wanted to make it easy for Dr. Butterfield on this. So she just has to have the dialogue with me. So in any event, this is going to be a quick run through of kind of the interesting history and some of the issues within cancer vaccines. But before we get to that, we have some poll questions for the audience. So we can't vote, so audience, please vote. You know, we have three questions here, three big picture questions about how important, will vaccines be, where will they best be deployed, and kind of the neoantigen question, the individualized vaccine approach versus some type of off-the-shelf approach. Hopefully some of these questions will come back to haunt us. We're at the end of our dialogue and taking Q&A with the audience. All right, so let's move into, if it moves for me, there we go. All right, so do you take the long road? That's not a question only for cancer vaccines. That's a question for much biopharma drug development. But nevertheless, cancer vaccines have had a very long and checkered past, as Lisa indicated before. This was a review from back in 2003 by Dr. Finn, clearly kind of talking about that gulf between kind of the excitement, the preclinical data, and the realities of how clinical development has gone on in the past, frankly, about 30 years, at least from the early 90s when some of the first work with anti-idiotype vaccines from Larry Kwok and others. All right, so the ignorance of experts. So for all the knowledge and expertise we bring to bear in trying to bring cancer vaccines ultimately to benefit patients, as I said, there have been a lot of travails, and I did have the honor about 15 years ago when we were first doing some of our early immunotherapy and cancer vaccine work, way before the approval of the checkpoints, to give a talk at Aventis Pasteur. And frankly, some of the same issues that were germane then are still relevant now in terms of, how do we do this? Are they cell-based? Are they specific antigen-based? Are they peptides? Are they, live cell dendritic cells? Do they get delivered via a plasmid or a virus? Are they, a mix of pre-specified, predefined, tumor-associated or specific antigens, et cetera? So those questions are very much with us today as they were 15 years ago. So for all the kind of travails, to go back to the title of the session, being kind of the false hopes, the modest wins, the many stumbles, basically in the US, the one true therapeutic approval we have is Provenge. The question is, what has happened? What needs to change, and we're going to spend most of our time in discussion with Dr. Butterfield about that, but also what has changed that perhaps is giving new hope and reigniting potential in the space. And I think we can, to paraphrase The Graduate, one word, not plastics, but checkpoints. And it really did help change and create this new field of what had been a backwater, of these lonely tumor immunologists into now this kind of headliner space of immuno-oncology that is so active a field, and investment, partnering, obviously, very significant sales in the space. Keytruda will be the leading drug, overall, not just in oncology. So, certainly checkpoints have given new hope to what we might be able to do with vaccines. Other things we learned along the way, and not just with vaccines, about following patients longer and the tail effect, and kind of what role chemo may play, because historically it was considered to be pretty much a detrimental thing. Certainly we'll talk about this idea of kind of where best to position vaccines. And we're certainly going to talk about kind of what types of adjuvants and antigens, as was in one of the poll questions, and the issue of kind of platforms. Is there a best, or is this going to be kind of a fit to purpose type of idea? The other thing I think we want to make sure, and anyone in the cancer vaccine field themselves certainly knows, that there's a lot of moving pieces here, which means there's a lot of IP, there are a lot of components that introduces additional risk for each of those, between the antigen discovery and what you're putting in, the platform that's for the immunization, any adjuvants that you may be using, other components that may be important to this. So a lot of moving parts. Again, not just complexity, potentially adding to cost, stacked royalties, et cetera, but also just, introducing additional elements of risk beyond the risk in just the clinical development studies and kind of the patient populations, et cetera. Fast. It becomes very much like a game of three-dimensional chess, in order to try to think of all these different components and figure out kind of which may be best in any given situation. So someone who's working on mRNA vaccines or oncolytic viruses, they need to think through what their system is going to benefit from most in terms of any inherent engineering or any concomitant agents, aside from checkpoint inhibitors. And of course, the choice of targets is very critical. And again, we're going to touch on that. And it's not just, kind of whether they are pre-specified or whether they're individual neoantigens. There are kind of elements in the choice of each of those, and how many to give, and issues of, kind of clocking up the immune system or having immunodominant issues, or also, more recently, the idea of kind of negative or inhibitory antigens, or what Genosha calls, inhibigens. And, even kind of more detailed recent expansion on kind of the idea of not only what these antigens are in Uber-Sahane's recent review, and there have been others, but also kind of the implications of the nature of those antigens in terms of perhaps where they would be best positioned in an individual's tumor or individual tumor type, as well as kind of what other things you might need to do to help make the patient responsive and recognize those as immunogens and mounted immune response. And we'll touch on these. So that question does remain, to paraphrase in this case, Sound of Music, so you can see far-reaching interest on my part, is that, like, how do you solve this problem of what to target and how to get cancer vaccines to the next level, right? Now, some of these targets, can be approached not with vaccines, and they are being approached with antibodies or ADCs or cell therapies of various flavors, or bispecifics. So the competition that vaccines face is not solely other vaccines. The other question, of course, is, what's the format of the immunization platform, mRNA or self-amplifying RNAs or oncolytic viruses or peptides or long peptides, et cetera, or double autologous, which some people have done, right? Harvesting the tumor, making individual neoantigens, and then creating cell therapy from the patient, whether that's autologous DCs or something else, which certainly seems rather onerous and laborious. But if the outcomes were to be phenomenal, that might be justified both in logistics and cost. But to date, they have not been so much. And there are other aspects maybe we'll touch on, hopefully, about like heterologous prime boost, et cetera. maybe we should be doing that for our COVID vaccine boosters that are coming up in the next few weeks. We, of course, should restate the obvious, which is why we're even discussing this, and that is because, of course, checkpoints, as phenomenal as they are, they don't reach all patients. They don't have the same efficacy in all patients. Not even all patients can gain access to them. So trying to broaden the responsiveness of checkpoint inhibitors and the number of patients who are going to benefit from this immunotherapy approach, not to say that it will necessarily obviate the need for other approaches, either before, after, or concomitantly, meaning chemo or radiation or targeted agents of various other sorts. But just thinking about kind of where the positioning for cancer vaccines will be is going to be very important, and we will touch on that in our discussion. Another thing, of course, is that cancer vaccine companies, in order to try and get to the next level, to try and get these more clinical benefits seen and ultimately get some new cancer vaccines besides Provenge across the finish line at the FDA or other agencies, is you've got to get the funding. To get the funding, you've got to stand out against the noise. You clearly can do so well when you have great data, but often this has to happen in advance of data. So you're often relying on preclinical data or early signals. And that's really challenging in the immuno-oncology space because there is a huge amount of noise and activity with all sorts of mechanisms and modalities vying for attention. So kind of the where's Waldo analogy. They do remain an important part of the pipeline, of the IO pipeline specifically, as you can see, here in red. But that being said, whether from a standpoint of partnering or investing, there's a bit of a disconnect there, frankly, like there is, as I'm going to be talking about imminently, between the preclinical data and the clinical data, or between the immune data and the clinical outcomes in patients. So as you see here in this snapshot, the amount of activity in cancer vaccines is relatively small from a partnering standpoint. Granted, this only went up through late 2020, but still kind of makes the point. It's not like it's changed dramatically since then. But as I said, the pipeline is still, fairly active. Most of it to predefined types of tumor antigens or other approaches are certainly not specifically neoantigens. They're a smaller percentage, although they're a much larger proportion of, say, the more recent noise over the past, 5 or so years in terms of investing and what people are talking about. And we will certainly be discussing that. So this issue of, trying to get kind of respect for cancer vaccines, there are certainly some big deals that have been done, such as the Nykode Vaccibody Genentech deal. But when you look at various types of reports, not just some of the analytics that I just showed, but this is from a fairly recent Travails weekly update, you do not see cancer vaccines on here. You see all sorts of different biologics, soluble biologics or cell therapies, obviously precision therapies, etc., ADCs, but not vaccines per se and directly. So this need for getting some respect for cancer vaccines, of course, is going to be very dependent on kind of proving that they can actually have clinical benefit. As I alluded to, while I don't have this specifically for vaccines, Howard Kaufman's analysis of oncolytic viruses does raise a very similar point for them, and there is a big overlap between oncolytic viruses and cancer vaccines about this disconnect between the preclinical data or even the immune correlate data, which in many, if not most of kind of the vaccine clinical trials has been promising, if not outright positive, in terms of effects on generating cells. Certainly CD4, a little more challenging CD8s, but various types of other cytokine measures, etc. But that remains to translate into actual clinical benefit. Significant, meaningful. Obviously, outcomes are most important in terms of OS, but even reasonable ORRs. Now, there are signals being generated from newer analyses. There's a review where some of these signals and early clinical outcomes are promising. I hope this continues. We can probably point historically, and if I had more time, I would collect some of the materials I have that historically also show, again, very promising early small studies in terms of clinical outcomes that just fall apart once you get into larger randomized either phase 2 or phase 3 studies. So hopefully these early signals, can prove themselves. Most recently, there was data from Gritstone with their heterologous prime boost, and hoping that these early signals of efficacy translate and continue to translate into actual benefit. But it's a time where, not just for cancer vaccines, but for many other companies, cell therapy companies and others, where even positive data, is not being rewarded, or in fact is being punished because the expectations have been so high. So, and I'm sorry, I'm doing this a little slower than I intended, only because the slides are being a little slower. So my New York ability to speak quickly is being compromised by IT issues. I'm going to skip that previous slide. One more thing I want to say is that, and this is very important, and I hope we touch on it, Dr. Butterfield and I, is that it's not just, kind of what you're deploying for the vaccines or what targets or antigens you're using, but you also have to really think carefully about where you're going to go. We're going to talk about that at the high level, but even at a very specific granular level, where you will be positioned, where is the value proposition, where is the unmet need, where is there kind of the Boolean intersection between where the patients are that are going to have most need of benefit from some type of improvement above and beyond checkpoints, meets where it's clinically feasible to develop, and where the particular platform, whether it's a vaccine or something else, is likely going to be best fitted. So this is where we spend a lot of our time with clients thinking through. So that is it. Again, sorry for doing it. Well, maybe not sorry. People are probably happy I didn't do it at 78 RPM, for those who remember what that means. But now let's get into the Q&A. And before I do that, I just want to give Lisa a chance to kind of make any big picture comments after I've kind of run through some of these major topics across the field. So Lisa.
Super. Thanks very much, Jeff. Yeah, that's a great landscape analysis, and you've covered a lot of really important topics related to cancer vaccines. And I've been at this long enough to have been doing something really hot and really cool, and also something that shouldn't even be brought up in an immunology lecture as something being pursued because it's over. So to have been through all of that and still be doing cancer vaccination is a lot of fun, actually. So I guess one of the things I wanted to start with and sort of get dealt with is because I've been working in dendritic cell vaccines for a long time. And so why would you do dendritic cell vaccines after 200 clinical trials and no approvals for autologous DC vaccines? And you mentioned they're a little expensive and they're complicated. And I guess as someone who's been in the clean room making dendritic cell vaccines and who's run a series of trials about them, the cost is, inclusive of a leukapheresis to prepare a course of autologous dendritic cell vaccines is somewhere between $7,000 and $10,000 a patient. And so when I think about cost, and you think about TIL and a year of checkpoint blockade, that's actually not a lot of money. And then when you think about the complexity, you probably do need a GMP suite in order to make these cells from patients and knowledgeable trained staff to make them. But it's 7 days start to finish. So when you think about TIL and preparing TIL over, weeks and months, and engineering cells and other things, that the notion that you put the cells in the flask and you come back a week later and you wash and load your vaccine. So when you think about complex manufacturing, I don't think dendritic cells are as complex as a lot of other autologous therapies. And when I think back to one of the dips in the interest in cancer vaccines, there was this opinion piece from the surgery branch and Steve Rosenberg's group telling us that it doesn't work, and stop doing it. And so what those of us with the rose-colored glasses, first we read that with, great horror, that we were wasting time. But the other thing is, when we read through and they had done an analysis of all the different platforms, dendritic cells are the best. So, even people who don't like cancer vaccines when they do a literature search, find that it is a reliable platform. So, in defense of dendritic cells, I make those opening statements.
Well said. We do have an N of 1 in Provenge that is autologous and pulsed with a PAP GM-CSF fusion protein. So we don't know exactly how it works, and we could debate till the cows come home, if it actually, how meaningful it works actually. So, but to your point about logistics and particularly cost, obviously, some of these approaches are going to be more feasible and more cost effective, let's say. Of course, we need to make the distinction between what something might cost, like the leukapheresis, versus what the product would be charged at the end of the day, like the $98K that Provenge came out at in its original approval in 2010 or 11. And then I think it is also important to think about, because the points you raise are important because they raise issues that a lot of people in the field need to think about, whether they're DCs or not, which is particularly for neoantigens, those, taking the tumor biopsy, doing the sequencing, the NGS, the TCR, whatever approach one is using, and then the algorithms, et cetera, that still does take generally a couple of weeks to do. And in cases where it is, even more complicated, like where people are doing functional assays to kind of confirm the nature of those neoantigens, that could take actually many, many weeks, even several months. And that's important to consider because, the more logistically cumbersome it is, the more costly we presume it will be. And also the higher the bar people will expect in terms of what it will have to do. So let's use this discussion then to talk about, we could segue right into talking about platforms, but I want to hold that for a little bit and just kind of back up a bit to the big question of, in your view, is there something that's really held the field back? Was it the lack of checkpoints? Did that just complicate everybody's efforts? They were kind of hitting their heads against a wall without the proper tools, or was there something else that we needed to be doing that we've learned since then, and that we are now improving upon as we go forward? Was it the lack of just the right tools?
I think it's a lot of things, and this also ties to a question in the chat about why those 200 fails for DC vaccines. Yeah. And I think part of it, I think it's multifactorial, and one of them has to do with the antigen targets, and that we went through this huge exercise with the NCI to go through all of the known antigens, and neoantigens have only been tractable technically for a small number of years. And so, historically, it's all been the shared antigens. And I think either your patient has the repertoire of T cells that can still, despite decades of talking to the tumor with that antigen in it, still has a T cell that can be amplified and respond to that, or you have a vaccine that happens to induce epitope spreading within that patient. So I think the successes we've seen have been these individual patients who happen to have the repertoire that can respond to a shared antigen until now. And so now we have a whole new spectrum, and that was a technology problem, and we're solving those technology problems. So one, I think it's antigens. And the other thing, we've seen, we do all our preclinical studies with healthy donor cells, and then you treat a cancer patient. And one of the things when I was running the core lab at University of Pittsburgh, we did a lot of dendritic cell trials. And so I got to see the readouts of everyone's trial, including my own. And we looked at the usual suspects of, things that we knew were important molecules on the surface of dendritic cells. All the dendritic cells express all of those molecules, and how much didn't matter. And when we went from healthy donor process development cells to cancer patient vaccines, those were not the same cells anymore. And since then, now we profile those cells and we see melanoma patients don't translocate enough kappa B to the signals that healthy donor cells do. And the molecules we were tracking, which was 3 flow tubes to get all 9 molecules, now we're looking much more broadly, as my cat reminds me. And
mine may jump up at any moment.
And so, now we're starting to find out what does matter, how much is on the surface. And so, how much, IL-12 P70 heterodimer do you need? How much ICOS ligand do you need? And things that we weren't able to look at. So I think we were able to profile more deeply to find out what the signals are. And, now we have technological tractability over a broad array of antigens that are going to activate a much broader repertoire of T cells that have had much different interactions with the tumor, truncal versus branch mutations, for example, over decades.
Yeah, great. So, and I'm glad you referenced, that the seminal Mac Cheever paper we often have used for all sorts of work over the years, but so let me ask the very pointed question then. Should people not be pursuing kind of those top-rated, tumor-associated specific oncofetal antigens? Should everyone be pivoting and focusing to neoantigens?
Well, we've vaccinated people with these shared antigens, these old-timey shared antigens like MART1, and all their tumor melted away. So we know that that's possible, and we've vaccinated people with neoantigens and their tumors didn't melt away. So I don't think it's all, there's I know we'll dig in a little more on neoantigens and the pipelines are all different, and different neoantigens are, people come up with different ones, and then, you have to decide what you're going to do with them to get them into the patients.
Well, it seems like we have this interesting situation. I mean, I did that very pointedly, creating that false dichotomy in the question, but we have this interesting situation where we have the long history of kind of the tumor-specific antigens, certainly, sporadic or subsets of patients in the many trials that actually had very good responses to those. And yet, as you just said, we have this new tool of neoantigens where, most people seem to be interested in now, and yet, for many of the studies published to date, either, certainly have minimal clinical benefits. I mean, they're starting to change a little, but, if you go back to like the original Neon and BioNTech publications, and those were mostly CD4 cells over CD8. So again, it seems like, there are things arguing in favor of both. Is anyone doing both together?
Well, there was the Amatic study, because you made the important point at the time it takes to identify neoantigens. And so when a late stage patient comes in, they had the protocol of let's vaccinate now with a shared antigen relevant to the tumor type while we identify neoantigens. And then we come in and we boost or, prime, depending on, with those neoantigens. And so I thought that was a terrific approach to testing both. And I think the central mechanism that we're after is epitope spreading. And if you get this, monoclonal response against one shared antigen that's been in the tumor, talking to the immune system, suppressing T cells, for a decade, that's not going to really cure people, just like a single T cell receptor to a single epitope and a single MHC. That's a tough road to hoe immunologically. But if you can initiate that process within the tumor of the tumor being able to now release antigens and have new rounds of priming, that spreading is, I think, no matter what you start on the front end vaccinating with, if those T cells are in that patient and able to initiate that process, you've won regardless of what your starting antigen was.
Yeah. And actually, I am aware of a number of other, I think BioNTech also has their kind of fixed vaccine to and then the kind of an individualized neoantigen. Yeah. And presumably they would want to do both, kind of buying time with the shared while they're kind of building out that individualized one, which makes a lot of sense because I think, I think that was implicit, use that word a lot today, in some of the things I raised in my slides about, kind of the timing, and maybe this will be the segue into talking about that, is like which patients do you pursue? Because certainly part of the challenge, probably historically, aside from some of the things we've already talked about, like lack of checkpoints, was, if you're going for a really advanced patient, aside from their being beaten up, at least historically, with chemo, and we'll talk about that too, they have very aggressive, in most cases, very aggressive kinetics of disease. And, it does take time to mount an immune response, whatever you may be using. So that does kind of raise the question of, and maybe we'll just leap to that right now, is like thinking about the best way to clinically develop and then ultimately position these for patients. And is the standard paradigm that, pretty much most agents and mechanisms and modalities still pursue, which is, late stage, later line, advanced stage patients, is that where cancer vaccines should be developed?
On the one hand,
figure they give you a low ball.
Yeah, I think, the surgery branch, again, has taught us that even bulky late stage disease can respond to an effective T cell therapy, in their case, adoptive T cell therapy. And in this discussion, this therapy that that is induced in vivo by the vaccine. And then my Pittsburgh colleague Olya Finn, has really done a lot of wonderful work looking at that pre-malignant stage, pancreatitis, longtime smokers, people with high-risk disease who perhaps, that maybe that's the role for the standalone vaccine, is that patient with high risk who's probably going to progress because it's not, it's certainly not toxic to receive most of these cancer vaccines as a standalone. But, that may be the place to at least get the immune system while it's still healthy, ready, so that to prevent that pre-malignant stage from going to full malignancy. So, and I know that she's analyzing some studies. Her initial study did show, though, that even in the pre-malignant stage, you have myeloid-derived suppressor cells that can inhibit even a vaccine at that stage. So there's still some caveats about the early stage. We know it's possible in the bulky stage, but in the bulky stage, there's so much else that's gone wrong that, maybe that's the adoptive cell therapy stage that, is really the wisest. So the cancer vaccine in the middle, I think, is going to be important for the patients who need it, and they're the patients who don't have the hot tumor yet. If they've got a hot tumor, we just have to amplify that and diversify that and we can do that with different checkpoint blockades as a standalone. But if it's the cold tumor, if you're not detecting those cells, assuming we know what cells we're looking for, then that's the patient who really needs to have that immune response primed and then come in with something that can amplify that.
So yeah, that sweet spot, what we call the lukewarm tumors, at least from a clinical development standpoint, might be a little retractable. Got a couple other questions that have come in, which are relevant to this discussion about kind of the antigens, neoantigens. One is about how much accuracy or robustness is needed in that neoantigen. So I assume that's a function of like, how good do these have to be at actually identifying things that are true immunogenic neoantigens? How many will it have to be? And what else will be necessary to get that epitope spreading? And I don't know if there's a magic number or even a magic answer to that right now, but I'm curious to hear your thoughts.
So far, it seems, more is better. And because no one wants to take the time to validate their neoantigens, they want to do the sequencing, do the predictions, synthesize the peptides and go. They don't want to then, look to see if there are T cells. They don't want to confirm natural processing and presentation by APCs or by tumor presentation, just make them and go. And so then, more is better. And Craig Slingluff at University of Virginia has shown with shared antigens, you can give 12, you can give class 1 plus class 2 together, and you can get responses to all of them. So the data I've seen to date suggests that, within, under 20, you're not going to overdo it and you're not going to block the possibility of immune response to any one of them. So without that validation, on the one hand, more is better because you didn't validate your algorithm, and it probably won't hurt.
Yeah. Well, I'm glad to hear you say that about. I wasn't aware of that study from Craig, but we often do get asked that, or people suggest that giving too much will kind of gum up the works, to be very vernacular about it. And as for the point of not testing them, I did allude earlier to some companies, and they've suffered for doing this, have kind of done that kind of extensive functional testing to try and confirm or verify which of those are really the appropriate neoantigens. But that does take, again, not weeks, that takes months to do. I mean, it's not to say that that's not a solvable engineering problem in part, perhaps, but it's still, there's not often that amount of time, which then brings us back to that idea of kind of that bridging with a shared antigen approach. Let's see, another question's come in. All right, so the question is really about whether there's any difference in kind of cancer vaccines between what's been seen with solid tumors versus heme malignancies. And, I alluded at the beginning to Larry Kwok's work, which was for NHL, but, most of the work that I'm aware of, continues to be more heavily focused on solid tumors. But, I'd love to hear kind of your commentary on this.
Yeah, whereas all the CAR T success to date is heme malignancy, and so much of the vaccine work and some of that success has been in solid tumors, although, in this small minority of clinical successes. And I think it's challenging when your antigen presenting cell is the tumor cell and it's interacting with the immune system. I think that makes it a little more challenging, but then I've seen some nice data, and I think it's AML in that minimal residual disease setting and preventing recurrence. And so I think that I think those types of studies demonstrate that there's no reason heme malignancies can't respond to cancer vaccination. And if we can get rid of some of that immune dysfunction with a chemo or something like that, get that down to a minimal residual disease setting, come in, and then, and even in hepatocellular cancer, just like in AML, there have been studies where if you vaccinate in that, adjuvant setting, there is a statistically significant reduction in recurrence rate and an increase in progression-free survival. So I don't see too much of a difference or a hurdle for heme versus solid tumors.
Yeah, I'm glad you actually mentioned kind of these settings like maintenance, et cetera, to kind of prevent recurrence, because I think that's kind of like the combination sweet spot between lukewarm tumors and then going to prevent recurrence after you've got your response, whether in solid tumors or conceivably in heme malignancies. Certainly there are some heme malignancies that are very, relatively indolent. So you would think it might make a good opportunity for a vaccine to come in and do something to kind of extend the duration of response or decrease the time to relapse. AML, on the other hand, tends to be very, very fast and aggressive. So that seems kind of challenging, but certainly a maintenance therapy in ovarian cancer or as you said, adjuvant settings. So
yeah, and I think, that's also a spot for patient enthusiasm. No patient wants to go home and just wait for recurrence. So I think there's, an opportunity for enthusiastic uptake by patients.
Great. So we have a question about combinations, and that's good because that's a topic that we've just lightly touched on but haven't gone into specifically. So, I would love to hear a couple of different things, your thoughts on. So one is kind of the actual way to best combine with checkpoint inhibitors. Is it post-progression? So you're kind of resensitized, you're adding on or keeping on the checkpoint, and how the sequencing should occur in timing between kind of when you give a checkpoint. And that may differ, say, between an anti-PDX agent versus a CTLA-4 agent versus the vaccine. So that's kind of one question. And then, of course, there's just kind of the standard of care questions like, can it or should it be combined with kind of chemo-only based regimens or TKI-based regimens or regimens that contain an anti-angiogenic inhibitor, given that there's some evidence that they may have some role in the TME. So let's start with the checkpoint one first, kind of how to think about that, and then talk about kind of the non-checkpoint.
Yeah, in my last melanoma study, it was just around the time that checkpoints were getting approved in melanoma. And so we had half the patients got checkpoint first, and then got a dendritic cell vaccine after, and the other half got a vaccine. So half got checkpoint first, half got, if they progressed, got checkpoint afterwards. And we saw that checkpoint first and proximal to the vaccine, because they progressed quickly if they progressed, that it set a higher baseline of antigen-specific T cells, but it didn't make the patients respond better. We did in vitro studies to look at the simultaneous signals of a vaccine antigen-presenting cell signal with PD-1 blockade, and that wasn't better. It didn't do anything for those T cells, but we had in patients who had gotten vaccine and then had progressed, got standard of care checkpoint afterwards, and that was either Ipi or Nivo or Pembro in that setting. And their vaccine-specific T cells in the periphery went 40 to 100-fold higher. And that came out about the time that Samir Khalif's mouse study showed that, that if you don't prime well first with a vaccine, then what you're boosting with the checkpoint is less effective. So a couple studies, have suggested it's vaccine first, then checkpoint for optimal T cell activation. But with so much standard of care checkpoint, our ability to do that is now less than it might have been, a decade ago. So I think we're going to have to look at that. And that may also be repertoire specific. There may be, cells that, may be more branch mutations, new, less suppressed T cells, they're ready. And also with the vaccine, you're only going to be activating a certain subset of the repertoire. The checkpoint blockade, would interact with the entire repertoire. So it's complicated. And, the signals I've seen suggest optimally prime well first and then treat with checkpoint. So that's my favorite, but it has to be tested.
So then let's just add another layer of complexity to make it that three-dimensional chest analogy. Then how might kind of chemo-based regimens kind of fit into this, given some of them, many of them kind of induce immunogenic cell death. And, if that's part of this kind of priming idea above and beyond whatever the vaccine may be directed to specifically.
Yeah, all the chemos are different. And yeah, if there's immunogenic cell death, then that could be an add. But, and also if you're coming in and you're clearing out the Tregs and the MDSC and a lot of the suppressive mechanisms and clearing out some, completely suppressed, terminally differentiated T cells, and you're sort of resetting the system. And then, after a time coming in with a vaccine, that might be perfectly good. I think there you might want to think about the age of the patient, because there, if you've cleared away all of those old T cells, what you'd like to do is prime naive T cells. And if the naive repertoire went away, after, 50 or 60, then you don't have the repertoire, you might have been better off boosting what was already there before you killed it with chemotherapy. So I think those are some of the parameters that we'll have to think about in those combinations and analyze the T cells for those answers.
Great. Okay. Another question that came in that I think is an important one to think about is, regardless, or maybe regardless of what antigens one's targeting, but certainly I would think this would be true of the neoantigens, how do we deal, or what level of concern is there for kind of the heterogeneity of the tumor, especially between the primary and the metastases, or, wherever you sample from, by the time you go back in, there may be further changes. So one way or another, there is selective pressure that, is occurring while we're prepping the vaccine and while we're giving the vaccine. So, I know that's why, more tumor-specific antigens has presumed to be better, which you referenced earlier, why, some magic number of 10 or 20 or more neoantigens. But, does that necessarily address this issue of kind of that recurrence, if those are going to be, if they have a very different profile? And is there a way to even address that a priori?
There's the shared driver mutant antigen, which may, it may be truncal and it may still be there in the metastases because RAS, for example, is so central to the biology of that patient's tumor that if we can get RAS to work, then, we might be able to then have a population of cells that really would still be there to respond to any metastases, because that is such a key driver to that patient's tumor. But I circle back to epitope spreading here, because that, I hope we'll get there. But for now, we're not there yet. And so we need to, think about what the modifications we need to make in the TME are to be able to enable that release of antigen in the right framework and the influx of useful endogenous antigen presenting cells, blocking the M2 phenotype, bringing in the M1 phenotype in order to have that second wave and create that autoimmune phenomenon of the first wave, is what you induce with the vaccine. And then that creates the opportunity for the second and third waves. And then you have a, polyclonal, polyfunctional repertoire that, has a much better shot at addressing metastases that come up later.
So it sounds like, to summarize that, the best approach might be one that, whenever the nature of the initial batch of antigens, maybe whatever they may be, that the key is really getting that epitope spreading and having the appropriate tools, which might be beyond checkpoints, to make sure that happens, and then letting the body more or less kind of be able to address then some of those heterogeneity that arises as the tumor evolves.
And I think, yes. And then, we've got this great, exciting new area that's becoming more tractable of tertiary lymphoid structures. And those, in the tumor microenvironment, are this positive signal. And now we're starting to really look at, those B cell components and the whole, that sort of structure, which is, there's sort of 10 different definitions, but we're, now that we can do all the multispectral imaging, we can look at all of those molecules and really identify these structures, because, that's, I think, an obvious tie to the opportunity to induce epitope spreading. If you've got these tertiary lymphoid structures adjacent to the tumor that are right there with the antigen, right there with the antigen presenting cells, and able to initiate new immune responses.
Great. And that also suggests, some of the things you just said, we know we have clients working in kind of those spaces, like Travails, who has a RAS mutations specific vaccines, or ENB Therapeutics out of NYU, which is actually, their agent seems to have an effect on TLOs. So that's really interesting to hear. But it also does suggest that probably other components, other combinations will be needed to help with that, like promoting the tertiary lymphoid structures, or helping promote that, the repolarization of the macrophages, or whatever that might be. So we got another question, which is a really interesting one, still on this topic of choice of antigens, which is, beyond kind of the standard methods for determining neoantigens. There are obviously a lot more being discovered that are rising from indels and RNA editing, and also this question doesn't even mention post-translational modification, like tumor-specific glycosylation changes or lipidation changes, or any of a number of other things. I know a few companies that are kind of focused on that. I think ScanCell for one, and maybe a few others, but I'd love to hear your thoughts about some of those let's call them amino acid backbone types of antigens.
Yeah. This is again, it's a wonderful revolution in technology that is enabling, I think, steps in cancer vaccination success by enabling us to identify all these other antigen types. And I'll circle back to Olya Finn and her long work on MUC1, and that this minimally glycosylated tumor MUC1 is responded to very differently than the normal heavily glycosylated MUC1. And so that makes that shared protein antigen a tumor-specific antigen, differential glycosylation. Lipidation, we're going to have to see the extent to which, those are processed and presented. Does that bring in NKT cells and other T cell subtypes that might also broaden the immune response from just cytotoxic CD8 that we've been so focused on, with the odd helper thrown in. So this is we've just haven't been able to look at the long read sequencing, is now enabling us to look at some of these other types of antigens. So I'm very excited about what we're going to learn in the next year or two about those, because as soon as we can break down, the antigens and the signals, then, it's not going to be dendritic cells forever, even in my own lab. It's going to be what the important signals are that those cells deliver, which then can be, moved to a synthetic platform paired with what we're going to learn about this whole new class of antigens over the next few years.
Yeah, great. All right, so we're moving into the home stretch. Here's a big question we haven't tackled yet. This issue of, we have a great diversity of immunization platforms. A priori, that might be really good, to just have these different things, and maybe they can play well together. But from your standpoint right now, aside from your evident bias towards DC-based vaccines, what are your feelings about kind of peptide vaccines, mRNA-based vaccines, oncolytic virus-based vaccines, et cetera?
And, peptides are, stupid, simple, and cheap, and absolutely immunogenic in, even in Montanide as an adjuvant. And they've shown the ability to generate, great T cell responses in the periphery. And they've suffered from a lack of tumor trafficking in the data I've seen. And so that's, so we need something else, something more. And, again, hats off to the mRNA vaccine folks who have, worked painstakingly to make that a stable, useful platform, because, the immunogenicity of that platform is really proven itself to be terrific. And, although we, may be saddened by our COVID our immunity that, then comes down after a few months. So, there's going to be room for improvement in the RNA nanoparticle platform, but nanoparticles are, are simple chemical constituents and combinations with different signals to drive the right type of immune response. That's all, I think, open-ended. So RNA, I think, is a great area that works now and will only work better. Dissection of the right signals to make them synthetic instead of cell-associated is a great future. And then, the oncolytic virus is also, I think, super important, because, what you want, you're trying to mimic a pathogen. And so if you start with a pathogen, now you've started ahead. You don't need to understand all of it yet. We will eventually, but you've triggered a lot of broad immunity. You've delivered a lot of signals, and you've delivered dangerous immune signals because of the virus and because of its replication. And so, there are viruses now, thinking of some collaborations with Cioxis in the past, where they can deliver that intravenously and have demonstration of tumor homing, anything, in the TVEC family that we've seen of intratumoral injections, that's easy, and that can generate a systemic immune response. So that adds pathogen signal and tumor microenvironment modulation, which are both super critical and things that these other platforms, don't do. So those are, I think, pluses about a lot of platforms.
So you may not be able to answer this. It just came to me. It'd be great if someone had the time and the money to investigate it properly, but just like many combinations, even historical combinations, we haven't really been able to study the proper timing, sequencing, dosing, et cetera. But my question is, I think everyone tends to think, oh, I have a platform and I'm going to focus on, a certain either tumor associated or specific antigen, or going to do neoantigens. But is there any reason to believe that certain targets, certain antigens might be better suited to certain immunization platforms?
Well, the synthetic long peptide needs to be taken up and processed by endogenous antigen presenting cells. So, we have to, those have to talk to each other to make that work. And yeah, that's certainly a concern that you need to have those matched well. I think there's probably, a fair amount of flexibility in that, especially if you're looking at full length antigens, then, all of them need endogenous antigen presenting cells. So if you have some sort of antigen presenting cell signal, even something simple like a GM-CSF, the old favorite, in the mix, with the antigen, then you're going to at least draw some APC, you're going to give them at least some sort of trigger, and you're going to have antigen in the same place. So I think there's some central paradigms that I would hope would carry through to reduce the complexity of needing to pair an antigen with a platform. I think it's more the form of the antigen and the platform.
Okay, great. I think we have time for maybe one more question that came in, and I'll see whether I have any more myself. And that is, is there a optimal route of administration for vaccines? I mean, people have done intramuscular, intradermal, etc. What's the data on that? Now, of course, some of that might be dictated by what the platform is, but like an oncolytic virus
Sure.
that you just referred to. But
sure, the early data with DC was intradermal was better than intravenous. We did a number of studies in Pittsburgh that looked, including intranodal, intralymphatic, and the heterogeneity of the patients outweighed the comparison, even with only 2 arms of the delivery. And so I don't think we even know that answer yet. There were early data suggesting, for melanoma, if it goes through the skin, then that goes to the skin draining lymph node near the tumor, and that's going to tell the T cell to upregulate, CLAW1, and a chemokine receptor that will direct it to dermal sites. And and that sort of didn't end up being supported, and you can give liver tumor vaccines intradermally as well. So if it needs to interact with endogenous antigen presenting cells, then a good intranodal or intradermal will get you that interaction with endogenous antigen presenting cells. And intratumoral, if you need to modulate the tumor microenvironment, that's where you need to go. But other than that, I think there's still the patient heterogeneity and the patient repertoire, I think, outweigh our ability to answer that cleanly.
okay, great. So then in our final 2 minutes, what are your, you don't have to name names necessarily, but what are your expectations for when the field might hope to see, some really encouraging data, if not an actual approval? I mean, is this something that, there are a variety of things that are advanced stage in terms of, their clinical development right now, but, if you were forced to be a betting person, are we talking about, within the next few years, or are we talking about a longer horizon before we actually see, the first new long coming cancer vaccine approval?
I guess if you're looking for a standalone vaccine in a late stage setting, I think we're a little ways away. But if you're looking for a standardized platform that works well enough that if you want to activate anti-tumor immunity to build on, this is the thing that will do that you can rely upon. If that sort of setting was approvable, then I think we've seen, especially in that progression-free survival setting, I think we've seen data that looks, clinical data that looks robust enough that we might have, that minimal residual disease, progression-free survival impact in the near term, and a more robust knowledge about neoantigen pipelines and platforms for that as well, because everything exploded, and now we're contracting a bit as we learn more about things that didn't work. So that then reduces that field to the things that are showing promise that will then go forward. So I think my spidey sense says a couple of years for the next thing in a straightforward way, and then a little longer for a therapeutic vaccine.
Okay. Great. Well, this has been a great discussion. I really appreciate your very thoughtful and insightful comments, Dr. Butterfield. Thank you for everyone who sent in their various questions. Thank you to our co-host, ShareVault. And there was my alarm, so we're right on time. So thank you very much. Thank you, everyone who attended. And again, the video will be available on demand, and anyone who wants the slides, even the fuller slides beyond what you can get with the video, let me know.
Thanks so much, Jeff. It's really been a pleasure. Always fun to have this discussion and conversation. Appreciate it.
Great. Thanks so much, Lisa.
Thank you both.
Thanks.